In 2010, DNA from a finger bone in a Siberian cave identified an entirely unknown human species. No skull, no skeleton, just a genome. For 15 years, finding a complete Denisovan skull was the holy grail of paleoanthropology. Then, in 2025, ancient DNA extracted from dental calculus linked a massive cranium, hidden since 1933, to that ghost lineage. The discovery did not just give Denisovans a face. New analysis now suggests our lineage's origins may extend beyond 1 million years. But there is a conflict the field still cannot resolve. The DNA evidence and the skull morphology point in opposite directions. In March 2010, a team led by Svante Pääbo at the Max Planck Institute published DNA results from a fragment of fingerbone found in Denisova Cave in Russia's Altai Mountains. The sequencing revealed a mitochondrial genome that did not match Neanderthals or modern Homo sapiens. Nuclear DNA analysis confirmed the divergence. The specimen's genome placed it as a sister lineage to Neanderthals, distinct from both them and from living humans. No recognizable skull or skeleton had been recovered, only a finger bone and later a few teeth. Yet the genetic distance was clear. The Denisovan genome branched off from Neanderthals around 400,000 years ago, and from Homo sapiens even earlier. This was the first time an extinct human population had been identified solely by its DNA. The announcement stunned paleoanthropologists. For over a century, species were defined by bones, not sequences. The Denisova cave discovery inverted that order. The field now faced a new challenge to find a complete Denisovan skeleton, or at least a skull, to match the genome. The search became a central obsession. Chris Stringer called the hunt for a Denisovan skull the holy grail of paleoanthropology. The few physical remains, teeth, a finger bone, a partial mandible from Baishia Karst cave, offered no full cranial profile. Each fragment was tested for DNA, but most yielded only traces or none at all. The Denisovan lineage was soon shown to have left a genetic legacy in modern populations. A variant in the EPAS1 gene, inherited from Denisovans, enables Tibetans to thrive at high altitude. But the people themselves remained invisible, known only from their genome. Fossil evidence lagged far behind the molecular trail. The gap between genetic data and skeletal anatomy widened as more Denisovan DNA was sequenced from cave sediments and isolated teeth, but no face or full cranium appeared. The absence of a skull made it impossible to reconstruct Denisovan morphology or to map their place in the human family tree using traditional comparative anatomy. DNA placed Denisovans closer to Neanderthals, but the few available dental and jaw fragments showed features more similar to Homo sapiens. This conflict between genetic and morphological evidence drove debate in the field. Some researchers argued that the molecular tree was correct, while others pointed to traits in the fossils that did not fit the DNA-based placement. As the years passed, the pressure to find a complete Denisovan skull mounted. The field waited for a specimen that could bridge the gap between sequence and skeleton. Without it, the Denisovans remained a statistical ghost, defined by divergence dates, not by face or form. The quest set the stage for a new phase in the investigation of human origins, one that would depend on linking molecular signatures to physical fossils and resolving the contradictions between gene and bone. The absence of a Denisovan skull became the central obstacle for understanding this lineage. Chris Stringer called the search for such a fossil the holy grail of paleoanthropology. Without a cranium, Denisovans remained an invisible population, defined by DNA, not by face or form. The gap was not just anatomical. It prevented researchers from linking genetic signatures to physical traits, reconstructing behavior, or placing Denisovans within the broader evolutionary tree using traditional comparative anatomy. The stakes of this gap grew as genetic studies revealed Denisovan DNA in living people. The strongest evidence is the EPAS1 gene variant, which enables Tibetans to survive at high altitude. This Denisovan-derived allele, present at nearly 87% frequency in Tibetan populations, alters the body's response to low oxygen by regulating erythropoietin pathways. The physiological effect is measurable. Carriers avoid the dangerous overproduction of red blood cells that causes chronic mountain sickness. The molecular signature is clear, but the people who introduced it remained a mystery. Only a handful of teeth, a finger bone, and a partial jaw could be tested for DNA. No skull had surfaced. Most fossil candidates failed to yield any genetic material, as mineralization and environmental exposure destroyed endogenous DNA. The few fragments that did produce sequences often showed high levels of modern contamination, requiring rigorous damage pattern analysis and Bayesian filtering to distinguish ancient from recent DNA. Even when DNA was recovered, it was usually mitochondrial, not nuclear, limiting the resolution of population relationships and admixture events. This forced reliance on indirect genetic signals, on adaptive introgression studies, haplotype mapping, and functional assays to reconstruct Denisovan impact. The evidence from EPIS1 and other loci confirmed Denisovan genes shape modern human physiology, but left their anatomy and evolutionary context unresolved. The field turned to alternative sources, such as dental calculus, to extract DNA and proteins. These approaches provided partial answers, but could not replace a complete skull. The lack of a cranium made it impossible to test whether Denisovans resembled Neanderthals, Homo sapiens, or a separate lineage. Morphological analyses of available jaw and dental fragments produced conflicting results. Some traits aligned with Neanderthals, others with modern humans. This tension between DNA-based and morphology-based placement became a core debate. Genetic phylogenies consistently grouped Denisovans as a sister lineage to Neanderthals. Morphological studies, however, often found Denisovan specimens closer to Homo sapiens. Without a complete skull, the conflict persisted. The urgency to find a diagnostic fossil intensified as Denisovan genetic influence was mapped across Asia and Oceania. The field needed a specimen that could bridge the molecular and anatomical evidence, a face for a genome. The search for this specimen set up the next phase of investigation as researchers scoured candidate fossils for clues that could resolve the Denisovan paradox. The Harbin skull entered the scientific record by an unusual route. In 1933, laborers recovered the massive cranium from riverbank sediments along the Songhua River near Harbin in northeastern China. The fossil was hidden away for decades, passing through private hands and eventually stored in a local museum. It remained largely unknown outside the region. In 2020, a team led by Cheng Ji at Hebei Geo University re-examined the specimen. They documented its exceptional preservation and initiated a detailed study. Stratigraphic and radiometric dating placed the skull in the early Middle Pleistocene. Optically stimulated luminescence on the surrounding sediments returned an age of about 146,000 years, with independent argon dating on volcanic ash, bracketing the deposit and yielding a compatible range between 130,000 and 160,000 years. The context was a fluvial sand and silt sequence, typical of riverbank deposition in the region during that period. In June 2021, Jai and colleagues published twin papers in the Innovation describing the anatomy of the skull. High-resolution CT scans revealed a cranial capacity of approximately 1,420 cubic centimeters. The specimen displayed robust brow ridges, a massive occipital bone, and a low, elongated brain case. The parietal vault was expanded, and the combination of primitive and derived traits did not fit any known species. The researchers noted a thick brow ridge, large mastoid processes, and a flattened face. These features excluded Homo erectus and Neanderthals from consideration. The team proposed a new species, Homo longi, called Dragon Man, named for the Black Dragon River where it was found. The 2021 publications compared the Harbin skull to other Pleistocene fossils from East Asia, including the Dali and Geniutian crania, but argued that the Harbin specimen was distinct. G's group also observed similarities with the Shehi mandible from the Tibetan Plateau, which had been identified as Denisovan through protein analysis. This raised the possibility that Homo longi might be closely related to Denisovans, but no genetic data were available for confirmation. Initial attempts to extract ancient DNA from the Harbin skull failed. The bone had been exposed to water and variable conditions for thousands of years, destroying endogenous DNA. Standard silica-based extraction protocols yielded less than 0.01% human DNA, far below the threshold for reliable sequencing. Only modern human haplotypes were amplified, confirming contamination from handling and storage. These setbacks forced a shift in strategy. Researchers turned to dental calculus, the mineralized plaque on the teeth, as a potential source of preserved molecular material. At this stage, the Harbin skull remained a morphological outlier with no molecular anchor. Chris Stringer, a leading paleoanthropologist, described the Harbin skull as the most complete fossil of a Denisovan found so far. The specimen's completeness and preservation made it a prime candidate for resolving the Denisovan paradox, a lineage defined by DNA but lacking a diagnostic face. The Harbin skull's mix of archaic and modern traits placed it at the center of the debate over how Denisovans relate to Neanderthals and Homo sapiens. Morphological analysis placed Harbin closer to modern humans, while genetic models, if confirmed, would position it with Neanderthals' sister group. The field now faced a critical test. Could molecular evidence from the Harbin skull finally bridge the gap between sequence and skeleton? Cao Meifu's laboratory at the Chinese Academy of Sciences began with a fragment of hardened dental calculus, weighing less than half a milligram, scraped from the Harbin skull's left molar. The team applied a decontamination protocol using ultraviolet light and sodium hypochlorite to remove surface DNA. Chemical lysis dissolved the matrix, releasing a mixture of host and microbial DNA. Silica-based library preparation, optimized for fragments under 50 base pairs, generated a sequenceable extract. The initial sequencing run produced 1.2 million reads. Schmutzy software estimated that approximately 80% of the sequences were modern contamination, consistent with decades of museum handling. Damage analysis showed a 12% cytosine to thymine substitution rate at fragment ends, a signature of ancient DNA. Filtering by fragment length and damage signature isolated a subset of endogenous DNA. Consensus calling reconstructed a complete mitochondrial genome. This genome clustered within the Denisovan clade, matching the haplogroups from Denisova 3 and the Jiahe mandible. No overlap was found with Neanderthal or modern human reference sequences. The result was published in the journal Cell in June 2025, with Fu as lead author. In parallel, a proteomic team, led by Dong at the same institute, analyzed a 5 milligram sample from the same calculus deposit. Filter-aided sample preparation and tandem mass spectrometry identified over 1,200 peptide spectra. 87% mapped to enamel-specific proteins, including amylogenin X and enamelin. 3 non-synonymous substitutions in the enamelin and amelogenin proteins matched variants previously found only in Denisovan genomes. These amino acid changes were absent from all sampled Homo sapiens and Neanderthal proteomes. The proteomic data displayed deamidation rates above 70%, confirming ancient origin. Modern protein contamination was below 2%, based on the absence of bovine serum albumin and other laboratory markers. The proteomic findings were published in Science, also in June 2025. The convergence of mitochondrial DNA and enamel protein markers provided independent molecular confirmation that the Harbin skull belonged to a Denisovan individual. Both lines of evidence excluded Neanderthal and modern human affinity. The combined molecular results matched the age established by optically stimulated luminescence and argon. Argon dating approximately 146,000 years. The Harbin skull became the first nearly complete Denisovan cranium linked directly to the lineage by ancient DNA and protein evidence. This breakthrough allowed the field to move beyond fragmentary teeth and mandibles. Chris Stringer described Harbin as the most complete fossil of a Denisovan found so far. The dual omics approach, pioneered by Fu, demonstrated that even heavily contaminated or degraded specimens could yield decisive results when multiple molecular signatures are extracted and authenticated. The identification of Denisovan-specific protein variants in dental calculus, alongside a matching mitochondrial genome, converted a long-standing candidate fossil into a confirmed representative of the Denisovan population. With the molecular identity of Harbin established, researchers could now turn to the anatomical implications, how the skull's features fit with the genetic evidence and what this means for the placement of Denisovans in the human family tree. The Harbin skull provides the first nearly complete Denisovan cranium. Its cranial capacity measures around 1,420 cubic centimeters, rivaling that of modern humans. The skull displays massive brow ridges, a low and elongated brain case, and a broad, flattened face. These features set it apart from both Homo erectus and Neanderthals. Morphological analysis reveals a unique combination of robust, archaic traits alongside a parietal vault, more typical of Homo sapiens. The Harbin specimen stands as the best preserved Denisovan fossil, described by Chris Stringer as the most complete fossil of a Denisovan found so far. The anatomical evidence extends the Denisovan range well beyond Siberia. The Harbin skull was recovered in northeastern China, over 2,000 kilometers from Denisova Cave. Comparative studies highlight similarities with the Jiahe mandible, found on the Tibetan Plateau and identified as Denisovan by protein analysis. The presence of Denisovan fossils from lowland riverbanks to high-altitude caves demonstrates a wide ecological tolerance. Genetic markers and morphological traits now anchor Denisovans across East Asia, from the cold plains of Heilongjiang to the oxygen-thin air of Tibet. This geographic breadth suggests Denisovans were not a marginal group, but a dominant lineage spanning diverse environments. Yunxian II, a heavily deformed skull excavated in 1990 from the Hanjiang River Terrace in Hubei province, entered the debate after a 2024 digital reconstruction. Synchrotron, micro-CT scans and shape from silhouette algorithms restored its original cranial form, optically stimulated luminescence and uranium series dating place the specimen between 940,000 and 1.1 million years old. Morphometric analysis compared 104 hominin crania using three-dimensional geometric landmarks. Bayesian phylogenetic inference grouped Junxian II with the Harbin skull and the Baishia mandible, forming the longi clade. This clade appears as the sister group to Homo sapiens, not Neanderthals, based on cranial shape. Molecular clock calibration, performed jointly by Xiaobo Feng and Chris Stringer, estimated the Denisovan and Homo sapiens split at 1.32 million years ago, with the Neanderthal branch separating slightly earlier, around 1.38 million years. Chris Stringer summarized the result, saying the Junxian finding more or less doubles the time of origin of Homo sapiens. This claim, published in Science in September 2025, challenges the standard model of a recent African origin. The analysis depends on the Junxian II placement and the reliability of morphometric phylogeny, which remains debated. The field now faces a deepening conflict between molecular and anatomical evidence, with the timing of our own lineage at stake. Svante Pääbo, architect of the Denisovan genome, maintains that nuclear DNA consistently places Denisovans as a sister group to Neanderthals. Chris Stringer, drawing on cranial metrics from Harbin and Junxian, argues that these fossils cluster closer to Homo sapiens. The field now faces open disagreement between molecular and anatomical phylogenies. Moroccan fossils, previously assigned to early Homo sapiens, and Atapuerca remains in Spain, resist tidy classification by either method. Some specimens yield no DNA, others present ambiguous morphology. Stringer describes the situation as a standoff, where the genome tree and the bone tree refuse to align. Both camps agree that the current fossil and genetic record is incomplete. No Denisovan nuclear genome has yet been retrieved from Harbin or Junxian. Proteomic markers, though distinctive, cannot resolve admixture or population structure. The next phase will require targeted recovery of DNA from deep bone or tooth cementum and expansion of morphometric datasets across Eurasia. Researchers are also refining protein reference panels to test for convergent evolution. The question remains unresolved. Does DNA or morphology provide the truer map of human origins? The answer awaits new fossils, new sequences, and new analytical tools. A complete Denisovan skull was the holy grail of paleoanthropology. Chris Stringer calls the Harbin skull the most complete fossil of a Denisovan found so far. DNA still places Denisovans closer to Neanderthals, while morphology links them to Homo sapiens. The Junxian specimen more or less doubles the time of origin of Homo sapiens. Each new genome or fossil redefines the human story. The search for our beginnings is far from over.